Here's how to assemble the flow cytometer. First, a flathead wrench is needed to open up the packing boxes. Once the main part of the machine is taken out of the box, tear off the plastic wrap around it and remove the foam. Then carefully disassemble the sampling needle protector. Remove the remaining foam, then reassemble the needle protector. Take out the power cable from the same packing box. Plug its rectangle-like end into the power outlet on the bottom right of the back of the flow cytometer. Take out all the component boxes from another packing box. One box contains liquid storage station and connecting tubes. One is test tube turntable. The rest 3 boxes are desktop components. Take out the computer screen, the screen holder, and base unit. First, fasten screw on the bottom of the screen stand. Fix the screen on top of the stand. Take out 3 corresponding cables and plug into the correct ports below the screen as shown. Then plug the rectangular ends of these three cables into the ports at the back of the base unit. Now, put the flow cytometer next to the computer screen and the base unit. Find a white box named USB 2.0 to DB9 adapter cable from the component box. Take out two cables inside the box and plug them into ports called RS232 and USB serial respectively. Please remember to fasten the screws on the end after plugging in the cable of RS232. Then plug the other side of the two cables into the back of the base unit. Next, tear off the plastic wrap around the storage station. Take out 4 black tubes from the same component box. Match the name tags on the tubes with the inlets on the side of station. Remember to match the red dots on the ends of each tube with red dots on each inlet, so that the tubes can be plugged in successfully. Then take out 4 transparent tubes with name tags and match them with the correct bottle. Plug the white ends onto the bottle tops. Place the liquid storage station at an appropriate distance to the main body of the cytometer. Remember, all the black tubes goes into corresponding left side inlets. Plug the gray ends into the corresponding right side inlets at the back of the main body. The final step of assembling process is to set up the auto test tube turntable. A 2.5mm hexagon head wrench is needed. Unscrew the two screws on the bottom plate, and then take off the plate shell. Remove the cap and pull out the connecting cable. Plug the rectangular end into the top part of the back of the turntable. Then carefully slide in the whole turntable and check if it's in the right position. Again, use the 2.5mm hexagon head wrench to fasten the screws on the bottom plate. The assembling process is now completed. Now it's time to check if the whole machine is successfully set up. Turn on the computer, switch on the power supply at the back of the flow cytometer. Press the power button and the laser button at the front. Click on the Site Cluster software on the desktop. The username is administrator and the default password is 123. Once logged in, click on Communication in the left side toolbar. In Visa Configure Serial Port section, change port into COM4, then click Open on the right side. Check on the bottom left if all ports are correctly connected and showing green. If there is any red light and error alert, check if all the bottles are filled with corresponding solutions, except the waste bottle, which should be empty at the beginning.